Okay. Uh, my name is AWET ARAYA. Are you hearing me?
Yes.
My name is AWET ARAYA. I am a master's student from Ankara University, Turkey. I have recently graduated from Department of Plant Protection. My topic is going to be about an important pathogen of cereal crops, which is COCHLIOBOLUS, SATIVUS, or Bipolaris sorokiniana. The fungus, the nature or biology of the fungus is something different from other microorganisms. The fungus Bipolaris sorokiniana, synonymous Helminthosporium sativum, Helminthosporium sorokinianum, telomorph COCHLIOBOLUS sativus, has become an important pathogen in many regions where barley and wheat are commercially cultivated. The pathogen is distributed all over the world, including Turkey, and it is more aggressive under conditions of high relative humidity and temperature associated with improper soil fertilization. So this pathogen can cause many diseases in plants, in crops, such as barley and wheat mainly. And also it affects some other minor cereal crops, but its impact is not that much. So it can cause diseases such as spot blotch, seedling blight, hair blight, and kernel blight, in which the first three diseases are the most economically important forms, and they are highly dependent on environmental conditions. Thus, diseases bring worse effect on different growing seasons of the major cereal crops, particularly wheat and barley. And the impact of crop health, disease severity, and environmental conditions on yield loss is complex, and hence it is challenging to manage this disease. Common root rot and seedling blight are among the most common diseases which are caused by this pathogen. They are important diseases of barley and wheat. Losses from this root rot and including seedling blight have been determined in many countries. For instance, in Scotland, COCHLIOBOLUS sativus, a seed-borne inoculum in barley resulted in loss of 15%. And this figure shows decreased yield per head and also reduction in stand establishment. Even in some other countries like Canada, Brazil, and in other parts of Australia, Brazil, this pathogen is dominant among fungi causing common root rot from soil-borne inoculum. The loss mostly could be due to a reduction in total number of heads per plant. However, when soil moisture is not a limiting factor, though infected plants are only able to produce few number of tillers, they may compensate the yield by giving increased number of kernels and eventually to increase production rate. Loss of the disease become the worst, particularly in seasons of less soil moisture content when plants are grown under insufficient water supply, especially in those semi-arid areas, semi-arid regions like the sub-Saharan regions of Africa. This disease is highly prevalent and it is more devastating. Fourth bullet, this is another disease which is caused by this pathogen, which is Bipolaris sorokiniana, one of the most important foliar disease limiting wheat production in warmer, non-traditional growing areas. Spot blotch can be easily seen in places where barley and wheat is grown. However, it causes significant loss only in areas with a warm, humid climate and is not a problem of great concern in areas with semi-arid conditions. Yield loss are exacerbated, especially when the farmers practice low inputs and late sowing dates. When the farmers practice late sowing date, this can give the chance for having infection from the soil-borne pathogens. In Mexico, when infested plots compared with chemical protected plots, it was reported that the infested plots produced 43% less than the protected plots. In Nepal, as on-farm studies indicated, crop loss as high as 16% has been found, and losses from Bangladesh has been reported. Biology of the fungus. Like many other fungi, COCHLIOBOLUS sativus reproduce both sexually and asexually. And this pathogen makes it different from other pathogens in that this fungi can be reproduced both sexually and asexually. And the asexual reproduction of this pathogen can bring to numerous spores that can infect the plants at different stages. And the fruiting structure of this pathogen is called Cerotecia, which bears many spores. The fungus reproduces sexually by forming a clave-like structure called asci. Asci give rise to amorphous, uniformly filamentous, and spirally tightened structures called ascospores. The mycelium of the pathogen is characterized by deep grayish-green to brown colored. When the fungi put under a culture medium, it produces numerous conidiophores, which may be in colony or singly. And the conidiophores contain different septa, in which the one structure is different from the other structure. Conidia are ovoid to long olive brown and with circular ends and contain up to 10 septations. Isolates of the pathogen are highly different in their virulence. Here, as we can see in the picture, it represents the growth type of the fungi when it is placed under culture medium. Here, the light color and dark colored. Most of them are dark colored colony. It can be produced singly or in colony. And as we can see the lower picture, it explains, it describes about the nature of the growth. As we can see, some of them are smooth growth and some of them flabby appearance. Here is the structure of conidia, which is produced by Cochhliobolus sativus. It is oblong, ovoid, and having circular ends. And each conidia is having different septa. It is divided into different walls. Here also the olive-colored conidia, which is clearly seen. It has different septations. Here, the conidia is usually produced through a stalk-like structure called conidiophores. Symptoms. The symptoms which are caused by this pathogen include dwarfing with discoloration or brownish of the roots and the coleoptiles are the most common type of symptoms which are caused by this pathogen. And under favorable conditions, lesions elongate and coalesce, and eventually can be callous and increase the size of the symptom, and it may reach up to complete days of the plants. These are some symptoms which are caused by the pathogen: root rot symptom and coleoptile symptom. Stunting of seedling infected by the common root rot organism. Here also root rot symptom. Here when the pathogen is soil-borne pathogen, it can affect the roots and can cause distortion of the roots, and finally to the days of the root, which can be uprooted easily the plants even by pulling our hand. This also similar symptoms. Symptom on the root. Sometimes it forms a ball-like structure, and sometimes, most of the time, we found on it rotting of the roots. The spots appear on leaves and leaf shades at different stages of the plant growth. Leaf supporting shows up as minute uniformly brown lesions up to 1 centimeter in diameter, which may continue to become large and eventually coalesce into extended area of blotches. Lesions are round to oblong with a definite margin varying in size because of sporulation of the fungus. Older lesions are olive colored. Severely infected plants show complete drying of leaves and development of kernel and head blight under stable environmental conditions. These are the typical symptoms of the spot blotch. It shows reddish-brown colored lesions on the surface of the leaves. Here also some common symptoms which are caused by the pathogen. in different crops, in barley and wheat. As you can see, when we compare the symptom between barley and wheat, here in barley it is much light in color, but in wheat it is darker and more, it has more legioness. And here so we can see we can compare the healthy kernels, the healthy grains with the unhealthy grains. The healthy grains are normally white, but the infected grains show some kind of discoloration. Disease cycle. Since common root rot fungi has widespread distribution in soils of cereal cultivating fields, most plants within population exhibit some degree of infection. Besides cereal crops, the pathogen can also attack many grass species. The pathogenous source of inoculum is mainly from soil, seed, and host debris, and primarily survives for a long time as a thick-walled conidia. Correlation of spore population with disease ratings of wheat seedlings has been reported in greenhouse, but not in the field. A pressurized and dome-shaped infection cushions are first developed on the host surface. Prior to penetration, minute infection peaks raised from the structures. Infection from epidermis normally continues to the cortex and then to endodermis. As a result, destruction of the tissue will take place, and the infection sometimes can progress to the stem, and the vascular boundaries will become more severe. The severity of common root rot caused by Cochhliobolus sativus is highly influenced by environmental factors. For example, growth of fungi is favored by warm temperatures of 20 to 30 degrees centigrade. In addition to that, soil moisture affects infection development and its severity. Since the fungus has a wide distribution in agricultural soils where cereals are grown, their spread from field to field is not a problem of a great concern. However, elements that cause soil to relocate, such as water, wind, equipments, and machineries at the same time moves inoculum of the pathogen. The pathogen can also spread for long distance with unhealthy seeds. Crop rotation. Crop rotation is an important element of the disease management for this pathogen. It is an excellent cultural practice for controlling the different forms of the disease. It brings a good return in decreasing soil-borne inoculum. Since the pathogen is soil-borne pathogen, therefore crop rotation is an important measure of controlling this pathogen. Because of the presence of wide host range of the pathogen and limited alternatives for crop rotation in different parts of the world, control measures applied by this strategy do not always prove effective. There is also some drawbacks of this crop rotation. Plant stress limiting factors such as avoiding fertilization of nitrogen, shallow planting accompanied by proper tillage, in good soils, and management of other diseases may play an important role in decreasing consequence of the disease. Although resistant cultivars against root rot have not yet been developed, plants show different degree of reaction to the disease, particularly in areas where Cochhliobolus sativus is the most dominant pathogen. For example, Gateway, Melvin, and Oli are characterized as highly susceptible, whereas Centennial, Bonanza, and Clark are among the moderate resistant cultivars. Using certified seeds, especially produced in dry conditions, reduce the chance of seedling infection. Fungicides with broad spectrum have been reported to provide best result. Seed treatment application generally brings improved yield, but cannot guarantee later root and foliar infections. As in case of spot blotch, in regions where spot blotch is a dominant disease, foliar application of fungicides may become advisable to get optimum yield of the crop. However, because of the equipment of frequent applications, chemical treatment is not always economically achievable. Although fungicides could be used to control the spot blotch pathogen, the most environmentally friendly and effective control method is the use of resistant barley genotypes. Some studies which are done in Turkey. Turkey is one of the countries providing the highest barley production worldwide. However, the production is increasingly affected by the spot blotch disease caused by Cochhliobolus sativus. According to the studies done by Celik et al., Just a minute. Sorry, just a minute. Sorry.
Yes, yes, please. Is that kindly try to complete?
Okay. Yeah. Land races 6A, 13, 16, 17, and 27 showed low and intermediate. According to the studies, some land races showed good resistance to the pathogen, and some other minor pathogen land races showed virulent, showed susceptible to the pathogen. For example, land races 6A, 13, 16, 17, and 27 showed low and intermediate infection response to Hatay and Kastamonu isolates, respectively. Land races 3, 4, 5, 9, 10, 11, 18, 21, 23, 26, and 28 showed intermediate infection response to both isolates. Therefore, it was concluded that barley land races showing low and intermediate infection response to Cochhliobolus sativus could be used in spot blotch resistance breeding studies or can be planted in areas where spot blotch is common. Seedling spot blotch reactions of 25 advanced barley lines to 5 single spore isolates of Cochhliobolus sativus were also determined, while many lines report showing intermediate resistance, but none of them was able to show low infection response to the disease. The study clearly proved that presence of rich genetic variation among the barley cultivars that could be a good basis for managing spot blotch disease through further advanced breeding programs. The result of the work also implies that cultivars grouped as resistant and moderately resistant could be preferred in areas where there is history of high infection rates. Thank you. I wind up my presentation here. Thank you for listening to me. Thank you very much.
Thank you for your presentation.